All right, everybody. It's about noon according to my phone, and phones never lie, right? So thank you all for coming. So we actually have a special speaker today, Dr. Forrest Smith, and he's actually presenting to us from Auburn University. So Forrest, thank you so much for doing this, especially using technology and remote viewing and all that kind of fun stuff. So just a really brief introduction. Dr. Smith is an associate professor, and he's also the director of graduate programs. So I presume he's really good at teaching, and in particular about computer-aided drug design. This has been a request in previous years when we did our drug discovery and development lecture series, that we did not have any information about how to use informatics and modeling to really aid in developing new drugs. So I'm really excited we were able to add that to our curriculum this year. So with that, I'll give the mic to Forrest.
Thank you, Micah. I'm not sure if you had a chance to look at your email or not, Micah, but I had sent today's presentation to you, and there are a number of links in this that people may find useful, links to software if they wish to download to become more involved in computer-aided drug design. So feel free to distribute that however you might wish to.
Great, thank you. That's very helpful.
But my goal today was, in case you're not really involved in computational modeling, to sort of get you started. So I've titled this The Practical Guide, and that's sort of the idea. It certainly won't be a comprehensive presentation, but it should be enough to at least allow you to dip your toes into the pool of molecular modeling and get a sense of what's possible and what's not possible. So with that, just a little bit of an introduction, and of course, it's working. There we go. So a brief history. The use of drugs have been around for a number of years. The Ebers Papyrus was available in 1500 BC, and it documented the use of over 700 different plant-based products to treat a variety of illnesses, salves and lotions and elixirs that were utilized, and certainly of questionable value, but it's been around for a long time. And that general idea of sort of hit and miss theory for the development of medications sort of remained for a very long period of time. And that really changed sort of in the middle of the 20th century with the rise of organic chemistry. The ability to actually identify the agents that were responsible for the pharmacological effects, and the ability to begin to synthesize those drugs, or to at least, in the case of semisynthetic compounds, to modify the natural products themselves. Computers began to emerge in the late 1980s, and there was great hope that computer-aided drug design would have a major impact on the development of drugs. And that really was not the case. There was a lot of promise that was associated with that, but there was sort of a minimal outcome really associated with the utilization of those early programs. They certainly began to build the framework for the types of programs that we use today, which I would say are more useful, maybe still have not completely fulfilled the promise of even those early days. But we are certainly getting better in terms of our ability to predict things and explain things. And there certainly have been a number of different instances where computational chemistry has had a major impact on drug discovery. In the 1990s, utilization of computers was sort of circumvented by the utilization of high throughput screening and robotics, so that automation became a major component. And it really became a matter of, especially in the cases of big pharma, them beginning to screen the compound libraries that they had available over these number of years where they had synthesized compounds. It was, again, sort of a hit or miss kind of an affair. And that really continues to be a major focus in regards to finding new lead compounds. One of the things that really has come about is the idea of utilizing computers to make the idea of high throughput screening more effective, because in and of itself, high throughput screening is not a particularly effective way of finding new drugs. What computational modeling often does is sort of begin to narrow down the libraries that are going to be screened themselves. And that in and of itself can amount to a significant cost savings. Computer-aided drug design has continued to advance, and we are getting now, I think, some really exciting results where we're really beginning to get to the point where we can begin to make predictions on some compounds in regards to their abilities to bind and begin to calculate free energies of binding and relate that to the biological activity that we're seeing. So when we look at computational chemistry, we typically break that down into ab initio calculations. Those are calculations from the beginning, from first principles. These are calculations that are typically run on smaller molecules. The idea here is that you are trying as best as you can to solve the Schrodinger equation. Of course, we know that we really can't solve the Schrodinger equation for anything more complex than hydrogen, but certainly more advanced assumptions can be utilized to get us pretty close to being able to get reasonable results. Generally, in terms of drug design, we don't utilize ab initio calculations unless we're looking at very small systems, and typically when we're trying to, say, look at reactivity or look at a transition state. These types of calculations are very useful in terms of accomplishing those types of predictions. At the other extreme, we have molecular mechanics. Molecular mechanics really takes a very, very simplistic look at the model of the atom. It can be envisioned as viewing the atoms as being balls which are connected by springs. We begin to look at the way the molecule orients itself and the various conformers that are associated with that. Semi-empirical calculations are really in the middle. They will treat the electrons, which molecular mechanics generally doesn't. You can begin to look at molecular orbitals, for example. They make a number of additional assumptions compared to ab initio calculations, so they allow for us to begin to observe a larger number of molecules. Bigger molecules can be handled by semi-empirical calculations. There are a number of different packages. MNDO is a quite commonly seen package for carrying out MNDO calculation or semi-empirical calculations. But we're going to focus really on molecular mechanics. These are the simplest calculations since we're not really considering molecular orbitals. We really aren't considering the electrons except as it might relate to dipoles or ionized species. And for that, or because of that reason, we can begin to look at much larger molecules, something that would then be appropriate for modeling a biological system. I don't want to get too much in detail, but just the general idea of molecular mechanics, we begin to break down the molecule into the various forces that are acting upon these balls and springs that subsequently attach them. So what we're looking at here is really the relationship between two atoms as they come in closer contact with one another. And in this one specifically, this is the Leonard-Jones potential, where we're really looking at non-bonded interactions. So this really relates to the idea of van der Waals interactions and how that might have an effect upon the overall conformation of the molecule. Here, again, now we're looking at charged species, and just to point out that there are equations that will then allow us to calculate these values. We're looking at Coulombic interactions between positively and negatively charged species, or partially negatively and positively charged species. Then break that down further into the bond lengths, and the general idea that there will be, for all of these things, a optimum distance between two atoms in a bond, for example. And that's going to depend upon what the two atoms themselves are and the environment in which they exist. Then we look at bond angles. Obviously, if we're looking at a typical sp2 hybridized carbon, we're looking at 120 degrees as far as the normal bond angles that will occur around that functionality. And then finally, and the one that is probably varied the most when we begin to look at compounds beginning to interact, especially when we think about ligands, are the dihedral angles, and rotating about, in this case, really a carbon-carbon bond, but of course that can be any bond depending upon what part of the molecule you're actually considering. These are the things that are typically varied the most when we're, for example, trying to see how it is that a specific ligand may interact with a protein. Now, these are all important as far as establishing the general conformation of the molecule, which is certainly important in terms of the way it's going to interact with a protein or a receptor. But a number of these become important in terms of understanding how that ligand may interact with the protein itself. In other words, there may be van der Waals interactions between the ligand and the protein, and there's going to be an optimal distance where that interaction can be maximized. The same sort of situation arises for the coulombic interactions, so that, for example, if we have a negatively charged species, an ionized glutamate residue on the protein, we may have a tertiary amine on the ligand that would allow for an ionic bond to form between these two functionalities. All of these are important then as far as explaining not only the conformation of the molecule, but the ways in which molecules interact with one another. Now, one of certainly the early ideas of molecular modeling and molecular mechanics was the idea of trying to establish what the global minimum for the molecule might be. The idea at that point was that it was the most stable conformer of the compound which actually bound to the receptor. And certainly that is a component, but assuming that the energy of the ligand is not too high, it's generally not necessarily true that it is going to just be the global minimum that is going to interact with the receptor. But we need to be mindful of this just in terms of beginning to calculate conformations. So the way these programs typically work, if you draw a ligand into one of these various force fields that are used in order to calculate the relative energy of any particular compound, is it will begin to try, assuming you direct it to do this, to minimize it, to try and minimize the interactions that might occur. It will try and create the most optimum bond lengths, the bond angles, and the dihedrals, as well as considering any coulombic interactions or van der Waals interactions that might occur. When that happens and it tries to minimize the conformers, it really sort of depends where on the potential energy surface you are as far as the input structure is concerned. So, for example, if we were somewhere here and we're just looking at a very simplified form of butane, for example, so we have eclipse forms, various gauche forms, and then the most stable staggered form here. But there are certainly high energy intermediates between these, in this case, the global minimum and just a minimum here, where if we're rotating about these carbon-carbon bonds, the central carbon-carbon bonds, we're going to increase the energy because of the van der Waals repulsion that's going to occur as these groups actually get too close to one another. So whether or not we will actually get to the global minimum or not really sort of depends upon where we start as far as our input structure is concerned. The way these programs typically work is that, for example, if we started here it would minimize, and it would minimize until the calculation converged down here, and it would tell us that the minimized form of butane was actually the gauche form, which we know is not true. But in most cases, they won't go back the other way. They're really designed to minimize the energy, but they're not designed to get over energy barriers. And that becomes a problem not only as far as the conformation of the molecules are concerned, but just in general, the ways where we're going to minimize the energy of the overall system if we're thinking about these molecules interacting with other molecules, for example. So one of the major things that came about was ways that we could get around this, finding these local minima and begin to find the global minima. And there are a number of ways of accomplishing this. There are systematic searches where we take really all of the torsion angles that are present in the molecule, and we systematically rotate every one of them through a set number of degrees, for example. And this is certainly doable in the case of something like butane, but as the molecules become much, much larger, it really doesn't become practical. We would really be calculating forever in order to try and find the global minimum. There are other ways of accomplishing this. This is the so-called Monte Carlo methods are quite popular. And really this is just the idea of randomly sampling the dihedrals and then allowing the minimization to continue. So that if we take different starting points and we do this enough, we can really begin to map that potential energy surface and have a good chance of reaching then the global minimum. In the case of simulated annealing, typically what we're doing, and this sounds a little bit weird, but we're applying energy, or it's generally referred to as heating the system up, so that this allows the system to have enough energy to overcome the energy barriers that might be present. Typically after that happens, then we'll slowly begin to cool the system down, the idea being that as energy is taken out of the system, then we begin to move to more and more stable conformers, and eventually, if we do this enough times, we can again find the global minimum. And then the quench dynamics is a really similar sort of idea. We, again, heat it up and then begin to utilize the various high energy forms, do that enough, and then minimize them, and that eventually we will reach the global minimum by that mechanism. So this idea of just sort of looking at rotation about dihedrals in order to look at a global minimum, just wanted to give you sort of a little bit of sample of some of the things that we were involved with and where this sort of became important. A number of years ago, we had investigated the idea of incorporating some of the key features in anthromycin and some of the duocarmycins. Both of these compounds are minor groove binders. They bind within the minor groove of DNA. They've shown interesting activity as anti-cancer agents. In both cases, they form a covalent bond with residues within the minor groove. They're a little bit different. Anthromycin actually forms a covalent bond with guanine, which tends to project out from the minor groove. The duocarmycins actually form a covalent bond with the N3 atom of adenine. In the case of anthromycin, it really involves this carbanolamine, which will lose water, and the imine functionality is then attacked by the guanine nitrogen. In the case of duocarmycins, the N3 nitrogen acts as a nucleophile, attacking the cyclopropane. Double bond, or the bond, electrons move in, and this ring will then subsequently become aromatized. Our hybrid molecule ended up looking something like this, and we had this tested in the NCI screen at the time. It came back with some interesting activity, close to nanomolar cytotoxicity associated with it, and went on to the mouse xenograft assay, but at that point it kind of petered out, and we didn't really do too much of anything with it for some period of time. The problem with the compound was, or what we envisioned the compound's problem was, that it really didn't allow for additional functionality to be attached to it so that we could increase the specificity for specific sections of the minor groove. So the idea was to simplify it, to remove this terminal ring here, this piperidine ring, leaving us the nitrogen. And then just based upon the way we had synthesized the molecule, we thought that that would allow for us to functionalize it either at the 3 position here or the N2 position here. And our idea was to utilize compounds similar to natropsin. Natropsin is a minor groove binder. It's where a series of drugs develop around this called the lexotropsins. And the idea is that because of their planar shape, you can kind of see it here, they're able to fit within the minor groove. And you have some flexibility within the molecules in the sense that if you have a pyrrole functionality, it will generally bind into AP-rich sequences. If you vary these to imidazoles, they will actually begin to recognize GC base pairs. So you have the possibility then of sort of guiding these molecules, this being the alkylating function, and this being the functionality that would then direct it towards a specific sequence of DNA. But the question then became, well, how is it that we're going to attach these 2 molecules together in order to allow this binding to occur such that the sort of concave nature of the molecule would be preserved and the reactive cyclopropane would be directed towards the interior of that curve. And we sort of, we did a bunch of different compounds varying the attachment point at either the nitrogen or at the 3 position, and then various ways of attaching the pyrrole functionality, just to indicate what we ultimately came up with. We found that if we attached it at the 3 position, this portion of the molecule would still bind, but the cyclopropane would typically be directed out of the minor groove, and as a result wouldn't be well positioned in order to allow for the alkylation. However, if we attach them at the 2 position of the pyrrole and the 2 nitrogen of the alkylating functionality, and then we varied this torsion angle, we found that this actually represented a minima. This is in terms of that torsion scan is about right over in this section. You can see that as it begins to rotate, the energy goes up, and around 0 degrees, it goes up significantly. Just to show you what that sort of looks like in 3-dimensional form and what that minima would actually be, you can sort of see it here. And these things are always a little difficult to envision when we're looking in 2D at sort of a 3D phenomenon. This sort of gives you, with some of the shading, gives you some of that sense, but you can kind of see it a little bit better here, where I've created a surface around the DNA. You can see how the molecule now fits fairly nicely within the minor groove, and it's a little bit difficult to see, but this little bump sticking up here, that's the cyclopropane. So it now seems to be at least fairly well positioned to allow for reactivity within the minor groove to occur. And if we look around here, we see we also have, in terms of the energetics of the system, we have some flexibility so that the rotation around this bond still can occur. Hopefully, even if it's not perfectly positioned, it can have some flexibility to allow the alkylation to subsequently occur. So, when we look at computer-aided drug design, moving on from this just general idea of looking at conformations, we typically divide those up into structure-based design and ligand-based design. Structure-based design, which is really what I'm going to focus on today, we have this idea of docking. And just to point out, when we're looking at docking, we're really looking at something more similar to a photograph. We're just looking at a single point in time, possibly not a particularly significant point in time, because generally in docking, what we're utilizing, at least in static docking, is in many cases the X-ray crystal structure. And the X-ray crystal structure is really the way that protein existed in the crystal form, not necessarily in solution. So there are a number of problems that occur that are associated with docking. Molecular dynamics is really more like a movie. In the case of the receptor is typically a solvent is present, so we can begin to allow for the effects of solvent, which are really, I think, in the early work had been dramatically underestimated. And it's what static docking really doesn't do. It doesn't really consider the solvent. And that is a major energy term when we begin to calculate the energetics of these types of processes. And then something that is fairly new, it's been around for a number of years now, but it's pretty exciting in terms of the possibilities that it offers, and that's this idea of free energy perturbation. Ligand-based design, that's typically quantitative structure-activity relationships. There are things like conformational molecular field analysis, the well-known CONFA, pharmacophore modeling, shape-based methods. The shape-based methods have become of some interest recently because of the possibility of doing these types of calculations using GPUs instead of CPUs, which allows the processes to speed up significantly. Okay, I'm just going to spend a fair amount of time looking at docking. To start out, we will typically get an X-ray crystal structure. That will generally be deposited in the RCSB protein data bank. There's a link for those, and there are quite a large number of structures that are available. You may have private data where you have gotten an X-ray crystallographer to solve a crystal structure for you, so you're the only one who possesses that knowledge. That's obviously a very ideal situation, because if something is in the protein data bank, there's probably other people working on it as well. In the cases where you don't have a crystal structure for your molecule, it's still possible to get meaningful data out of that. That's the so-called homology modeling, where if we have the primary sequence of a protein and it's similar enough to a protein that has been crystallized, we can begin to build useful models as far as docking experiments. And then it is possible, I don't do any of this, but they tell me it's possible to use NMR, nuclear magnetic resonance, in order to solve protein structures to the level that will allow for docking experiments to occur as well. Then the ligands, proprietary ligands, you may have those, you may have just a couple of them that you're interested in looking at and determining how it is that these molecules actually bind. But if that's not the case, and probably regardless of that, you're going to want to begin to look at databases to begin to try and find other molecules that might bind to the receptor or target of choice. And there are literally thousands of these real databases that exist. I've just mentioned a few here. FDA-approved drugs. This is a fairly useful one to utilize, just in the sense of looking, for example, for off-target effects. There may be existing drugs that are out there. You have now come up with the idea that your compound may bind to myeloperoxidase, for example, or some other enzyme of note. We actually have done a little work in this area where we're working with inhibitors of myeloperoxidase, and noticed that a drug that was utilized in the treatment of rheumatoid arthritis, tofacitinib, which is actually an inhibitor of the JAK-STAT pathway, seemed to have some similarity to compounds that were known to inhibit myeloperoxidase. So we've made some proposals along that line of creating a dual inhibitor, since myeloperoxidase is an enzyme that's involved in inflammation and rheumatoid arthritis, although there's a signaling pathway that goes on in therefore tofacitinib is used as an inhibitor of that signaling pathway, the JAK-STAT pathway, that there would be value in adding or increasing the ability of these compounds to act together, to act as both JAK-STAT inhibitors and also as myeloperoxidase inhibitors. Another one that I want to mention is zinc 15. Sorry, my light goes out from time to time because of where the sensor is. Currently has about 100 million compounds that are available in zinc 15. And these are all supposed to be purchasable. I will tell you that term purchasable is used rather loosely. Some of those compounds are on the shelf and you can buy them. In some cases, there's small chemical companies that have to make the compounds, and it may take you months to actually get them. But all of these, the really nice thing about zinc is that really all of these molecules have been prepared appropriately. They have been ionized correctly. The formats of the files are such that you really don't have to check them, and you still will get good data when you submit these to various docking calculations. Obviously, with this number of compounds, you can't go through and check each one of those compounds to make sure that the atom types are correct, or that you're looking at the correct ionization state for those compounds. These are generally considered to be good, clean compounds. There's also, you certainly don't have to download 100 million. There are various smaller subsets that you can choose that are more appropriate for what specifically it is you're trying to accomplish. And then there are virtual databases. Enamine has recently created something called the REAL database, which has 720 million compounds currently present in that. And by these virtual databases, I'm saying that these compounds are generally created by combinatorial methods. One of the problems with the virtual databases can be that even though they may have a variety of very interesting compounds in them, can these compounds actually be synthesized? There are ways to sort of push it in that direction. So for example, you can choose a number of different coupling reactions, Suzuki coupling or Heck reactions. Choose a variety of those where you can begin to put fragments together, and that at least gives you some general synthetic route into making the compounds that then make up the database. It's been estimated that of these 720 million compounds in enamine, 85% of them are thought to be reasonable in regards to synthesis. But this can be a problem as far as just looking at virtual databases. And we'll move on. So there are a number of steps that must occur before you're going to actually carry out the docking itself. You need to prepare the receptor. And I'll show you a little bit of an example of this later. You're going to have to add hydrogens, because when you get the crystal structure from the protein data bank, assuming that's where you got it from, it will not have hydrogens present. The hydrogen is smaller than the X-ray is, and as a result, we won't see hydrogens being present. You may need to remove the water. In some cases, not all cases, but in many cases, the proteins are crystallized with some molecules of water. And the reason that I have a question mark there is which ones do you remove and which ones do you keep? So for example, water bridges may be an important component of the way specific ligands will actually interact with the receptor. And so then which ones do you keep and which ones do you get rid of? That's always a question. You need to check the ionization, add the missing side chains and atoms. Yes, in some cases, there may be missing side chains even from the PDB files. Deal with metals and subunits. A number of docking programs have problems as far as handling metals. Subunits like hemes, for example, they may not have the accurate parameters for those, depending upon which one of these programs you actually utilize. And you generally are going to want to minimize it as well, because what is going to happen is you're going to add the hydrogens, and you're going to do some other things as well, and especially the adding of the hydrogens. We know what those bond angles are going to be, but we still have the possibility of rotating around the dihedral angles in ways that may, for example, optimize the hydrogen bonding that's going to occur within the protein itself. Finally, you will generally create the grid. Now, obviously, all this typically involves is you designating on the protein itself the area of interest, the specific binding site. After that, really, it's typically a matter of pushing a button in order to create the grid. But really what the grid is just a simplified version of the amino acids around the active site that would allow for interaction. It will be the way that the program will actually evaluate the various docking runs that are going to occur. And then you have to prepare the ligands. Again, I said if it came from zinc, it probably would be okay. But especially if you're creating your own ligands because you have a compound and you want to know how it binds, things that you need to be aware of is checking the ionization. Are there tautomers present, and are there atom types? And sometimes this is kind of behind the scene. Various docking programs have different ways of actually identifying what those atom types are. I mean things like, is it an sp3 hybridized carbon? Is it an sp2 hybridized carbon? What kind of nitrogen is it? Is it sp3? What's the geometry around it? For example, amide nitrogens are somewhat different. Okay. Typical time to dock, around 1 to 100 seconds per compound per CPU. It really depends how thorough you want to be in regards to exploring the potential energy surface, how much rotation around the dihedrals are you going to look for. Many programs offer you a variety of presets here, so that if you want to screen compounds very, very quickly, you can set to really not sample thoroughly. Then, if you get that down to a select number of compounds, you may want to spend a greater amount of time to more accurately reproduce what you think the binding may actually look like. And when I say CPU here, I don't mean in the sense that generally, for example, desktops will have a single CPU in them, but that CPU now, depending upon your computer, will generally have probably around, for a newer computer, 6 to 12 cores in it. And those cores generally can each handle 2 threads per core. So if it was 12, that would count as something like 24 CPUs. You can get compounds with actually computers with 2 physical CPUs in them that would be, with the newest and most expensive Xeon chips, close to 100 threads that you could look at. That time to dock, again, it's dependent upon the active site, the size of the ligands, and the number of rotatable bonds. Generally, when we're just looking at a static docking run, we're only going to look at rotating the bonds in the ligand. There are ways to allow the side chains of the protein to rotate. In that situation, you're looking at induced fit docking, and there you're looking at typically around 20 minutes per compound per CPU. So your ability to do really very, very large databases, it's going to be difficult. It's going to take a lot of computer resources in order to accomplish that. What are you going to ultimately get out of that? A series of poses for each compound. Typically, programs can redock ligands the protein was crystallized with very well. So in some cases, you have proteins that are crystallized with a ligand, and usually the first thing you want to do is you want to extract that ligand, because you know the answer here, and see if the program can redock it accurately within the active site. That should be angstroms. Generally, what you're going to see there for the root mean squared deviation is around 0 to 2 angstroms. Programs are generally able to do that. Now, bear in mind, the resolution of the protein is probably going to be around 1 to 2 angstroms itself. In other words, when you get an X-ray crystal structure, there's still some fuzzy areas in terms of where those atoms may actually be. You're also going to get a docking score. And this is what people really don't like. Docking scores do not generally correlate well with experimental binding data. They just will not line up very well. And I'll show you some examples of that in a little bit. You're also going to get a ranking of the docked poses. It's going to tell you, because it's going to give you a score, one of your compounds was best and one of your compounds was worst. But, of course, since the scores don't correlate with the binding data, the ranking doesn't either. So the question then becomes maybe, so what value is this? Well, I'm going to try and build a little bit of a case for it. Yeah, it still can be of some value here. So this was a project that I was involved in that involved inhibitors of histone deacetylase. Specifically, the PDB ID, all of the compounds within the protein data bank were given a 4-letter or number code that will identify it. This was for LY1. And what I've done here, I've removed all of the protein for you, because if you try and look at these things with all of the side chains surrounding them, it becomes very, very difficult. But this was in green here. What you see is the way that the ligand was actually bound within the active site of that deacetylase. You can see some of the issues that I pointed to previously. For example, the green 6-membered ring here is actually a phenyl ring, but because of the X-ray crystal structure, it's not going to assign bond orders. You also see that there aren't any hydrogen present. Remember, I said that hydrogens would normally not be present. So what's in green is actually the experimental ligand and the way it bound. In gray was the predicted ligand. So in this case, the program, we specifically utilized a program from Schrodinger called GLIDE, was able to reproduce this fairly well with a root mean squared deviation of around 0.44 angstroms. So at least that told us that the program could reproduce known results. And this just shows you the ligand interaction diagrams that and how similar they are. This is the experimental, the way that it was interacting. Histone deacetylase is a metalloprotein. It contains zinc. And this is showing you the way, again, we don't have bond orders because this is from the X-ray crystal structure. There's an interaction between the aniline nitrogen coming off of this phenyl ring and then the carbonyl that's a part of this amide linkage. Those sort of coordinate the zinc and sort of stabilize the ligand. And you can see this is actually the predicted, where we see the same type of interactions as well as some additional interactions as well. Hydrogen bonding with some histidine residues, and whenever you see an NH and it's showing a hydrogen bond to glycine, that's part of the peptide backbone that it's actually interacting with. But the primary interactions are really the ionic interactions occurring with the zinc, and then there's an additional pi-pi stacking interaction here with this phenyl ring. Okay. So this is some of the results that we've gotten from some work that we've been doing with cannabinoid receptors. There are 2 types of cannabinoid receptors, the CB1 and CB2. These are G protein couple receptors. They actually have agonist and antagonist forms. And this is some of the best data that we got as far as our CB2 agonist binding. Correlation coefficient of around 0.49. This would be the more typical results that we would see from trying to get a correlation between our predicted binding and the actual binding score. I think this got cut off, and in both cases, we're looking at PKIs against the GLIDE docking score in both of these. So we really don't see a useful correlation coming out of this. And this is really sort of typical of what you do see from docking studies. I will tell you that the commercial programs, depending upon where they are, they will always say that, you know, you get really good results, and they will often cherry pick their results. They will go to something like this or something even a little bit better. But I will tell you, this is really much more typical of what you expect to see from a docking experiment. So what value are these things, these docking studies? What docking studies are best suited for is compound enrichment. So they can really begin to sort out the active compounds from the really bad compounds, typically cutoffs of around 10 micromolar or more. Usually the programs can sort those out. Now it kind of depends upon the model that you're utilizing, and I'm not going to say that universally that is the case. The way that you typically will test this then is you will take your known actives and you dock those against either known inactives. It may be difficult to find a large number of inactives, and the alternative is to utilize decoys. And I'll just introduce you to this idea of the DUDE database. That's the database of useful decoys. What you do with DUDE is you take your active compounds, you submit that to DUDE, and it's going to give you back a database sort of based upon rearranging the functionality that is present in your active compounds in order to create random decoys. So rather than looking, for example, at just other drugs, it really is allowing you to get down to a further amount of refinement in terms of whether or not you're going to be able to utilize your docking runs to begin to sort out the actives from the inactives. So this is some of our results from 15 known cannabinoid 2 agonists plus 750 DUDE compounds and their ability to interact with the CB2 receptor. And there's a whole variety of ways of looking at this, but just to sort of simplify this, this is sort of an enrichment plot. And what we're looking at is if there was no enrichment, if the compound, if the program found our actives at pretty much the same rate that it found the inactives, it would more or less run along this line, the diagonal here. If it actually found the inactives to a greater extent than it found the actives, we would be over in this portion of the graph. And this was really about our best run that we could achieve. So we do see significant enrichment, and it gives us some confidence in terms of utilizing this model in order to look at screening a larger number of compounds to possibly begin to develop novel agonists for this receptor. There's not a lot of interest in this as possible pain treatments with some of the opioid epidemic that we've had. People are very interested in beginning to determine new pain medication. So just a couple of modeling programs for you to look at. Autodoc Vena and Autodoc, both of those are available on the Alabama supercomputer. You can download those for yourself, and they have a GUI interface. Autodoc Tools is available to utilize with these. Autodoc Vena has gotten some very good reviews in terms of its ability to reproduce those root mean squared deviations for known compounds. That's often the way that people will begin to compare these programs. Some of the commercial programs, Schrodinger, Molecular Operating Environment, or MO, are available here. Obviously, you pay bucks for these things. Typically what happens is that many of the programs that are present, not all, but many of the programs that are present in the commercial packages were really developed in academic labs, and in some cases they're free. But what these companies will generally do is really develop the graphical user interface to make these programs a lot more accessible and a lot easier to use. So just something to bear in mind. Now, if you don't want to do any of that, and you just want some docking information, there are docking servers. I've listed 3 here, and literally all you do with these materials, with these setups, is that you will put in your 4-letter number code for whatever your PDB file, protein data file that you're interested in, and then whatever ligands will be uploaded into the server as well, and it will take care of all of this in regards to preparing your ligand, preparing the protein, and then depending upon how big your database is, you will get it back in a reasonable amount of time. Obviously, as the databases become larger, it takes longer and longer to get this information back. I just wanted to mention very briefly the idea of homology modeling. Assuming you do not have an actual crystal structure for the protein that you want to be interested, you're interested in, if it's not been crystallized, you can still begin to generate useful models. There are different numbers in the literature that are mentioned. You typically want sequence homology greater than 35% before you can begin to create useful homology models. The cannabinoid 2 receptors that we were talking about, these are G protein coupled receptors. They can be especially difficult. They do exist both in the agonist and antagonist form in G protein coupled receptors, which are very often targets for drug development. Have the additional complication is that those receptors are actually embedded in the cell membrane. And there are ways of handling that, but that has certain important influences in terms of the way these molecules behave. Okay, now there are homology servers. Again, assuming you have the primary sequence for the protein, you can submit it to a variety of different servers. iTassr is quite popular. Swiss Model, which also has Swiss doc available, is also one that has been around for a number of years. Generally, these do not allow you very much flexibility. They will generally just take the primary sequence that you submit to them and spit you out a model for the protein that you have submitted. If you want more fine tuning, where for example, you know that a specific amino acid is important for binding, and the models that you're getting out of the servers don't really reflect it. It's putting it out of the active site, for example. There's a program called Modeler, which will allow you to do this, and then will allow you to go on and create the homology model itself. And then just finally, I wanted to talk about this idea of free energy perturbation. And this is something that begins to address many of the problems associated with static docking. It utilizes molecular dynamics, so the protein is allowed to move. And really, that was one of the problems that was associated with the static docking that we talked about before. It may simulate the receptors existing in the cell membrane. For example, one of the programs I'm going to mention to you, really, it's just a click of a button once you have your model created to put it in a membrane and then surround that with water. So literally, one button push will allow you to do that. It incorporates the solvent into the calculation, and that is a major issue. We don't typically think about that, but when we think about a molecule binding to a receptor, one of the major energy terms is the idea that what was occurring before the ligand got to the receptor, and that generally means that the receptor was occupied by water. And there is a lot of energy associated with removing that water from that receptor, and that generally is not something that is reflected in a calculation like a docking run. There are limitations, however, on free energy perturbation. It works best for solved crystal structures. So you really have to have a good geometry and a good starting point to begin to carry out these types of calculations. And it also tends to work better for congeneric series of ligands, ligands that really differ very little in terms of their overall structure. Now you can begin to build off this, so you can take a simple structure, you can add a methyl group for it, and begin to calculate the energy difference between these. And then once you have the methyl, then you can begin to build off of that a little bit more. So you can begin to move away from your initial structure to begin to sort of develop novel structures. But it is a slow process. This is the basic idea of free energy perturbation theory. We generally look at it this way, where we have a ligand that is surrounded by water, a protein which is also surrounded by water, and we're going to look at it docking into its receptor. The idea with free energy perturbation is consider another system where we have a change in the ligand, a different ligand. Again, it's going to be similar, but it's still very much related to compound 1 that we were looking at over here. And we look at its docking. And here's the basic idea. So that rather than calculating the difference in relative free energy and going from the unbound to the bound going that way, we calculate the energy of changing this molecule into this molecule. And in the bound state, we calculate the difference of converting this molecule into this molecule. Sometimes this is just referred to as alchemy. The general idea of doing that means that there's much less of a change in going from here to here, where in both cases the ligand and the receptor are solvated, and here to here, where the bound ligand and receptor are both solvated, than going the other way. And that tends to reduce the error within the calculation. So the difference in the relative free binding of going from or between G1 and G2 is going to be the same as the difference in the relative free energies of going from ligand 1 to ligand 2 and in the solvated form, and bound ligand 1 to bound ligand 2. They will typically talk about being able to reproduce binding affinities within probably, say, 1 to 3 kcals per mole. And this is just some of the data that you can see, which looks much better than most of the data that I talked about previously in terms of being able to predict the PIC 50 values and the experimental PIC 50 values. These things generally will run on GPUs. Typically, dynamics runs will run on GPUs. And generally, it's going to take about 24 hours to do a single compound. So again, you're not going to be able to typically do a large number of compounds, but within a very narrow range there, you can begin to get useful data and start to make really accurate predictions in regards to the way various ligands bind to proteins, assuming you have a good starting point. As far as molecular dynamics and FEP programs, I would recommend Desmond. There's a very good graphical user interface to Desmond. It's free of charge, and it really makes things very, very simple. The problem with Desmond is if you're not somewhat familiar with Linux, then that's going to be a problem because it will only run under Linux. NAMD runs under Windows and Linux. It doesn't have nearly as much ease of use as Desmond does. There is a graphical user interface, but it's not connected to the program nearly as well as what you see in Desmond. Just a few acknowledgements. 2 of my graduate students. Ms. Logan Neal has done a lot of the work on the cannabinoids. Mr. Mansour Turkey is working on the synthesis of some of those minor groove binders that I talk about. And we've gotten Department of Justice support more from a forensic standpoint. And then Ms. Neal has gotten a couple of fellowships recently that have been very useful in terms of some of her work. If you have questions, I would be happy to entertain those. But otherwise, thank you for your attention.
Thank you so much, Forrest. Really appreciate it.